So I wanted to review this process of the acellular infectious particles that are out there and just remind you again some of the material that you can find in the textbook, and I'll go over some of the other stuff. But again, just to remind you, here we can see the various sizes of the different viruses that are out there. And remember, these viruses have to be able to fit inside of the larger cells that are out there, so we know that they are going to be much smaller, and because of that they aren't going to be able to be visualized or resolved using your standard light microscope, because they're just too small below that 200 nanometer size where you can resolve your particles that are out there. So you can see that we have to use that electron microscope to be able to visualize these small particles. And just as a reminder, when you're looking at viruses that are out there, they basically have to have two things associated with them. They need to have a nucleic acid, and they also have a protein coating around the outside with capsomere proteins that form what's known as the capsid. And on the surface of the virus, you're also going to find your spike proteins that they're using to attach to the host cells. And in the bottom example, what we can see here is an envelope virion, where the nucleocapsid that you see in the top figure is going to have an envelope around the outside, and that's where your spikes are embedded. And when you're looking at this envelope here, it actually is part of the host cell membrane that's going to assemble around that nucleocapsid as the virus is able to bud off of the cells. Also, just to remind you, we do have a variety of different kinds of shapes to the viruses, and this can be used in the classification of viruses in some schemes that are out there. And you can see our standard kind of helical shape right here, where the capsomere proteins basically assemble right around that helical form with your nucleic acid on the inside there. You also have your icosahedral form right here, which has these regular kind of triangular faces to it. And then you have the last example, which is your complex viruses that don't fit into these other two categories, and they are going to come in a wide variety of shapes and sizes that are out there. Also, just to remind you, when we look at these acellular infectious particles, viruses have that nucleic acid with the protein coating, but there are other kinds of infectious particles that actually lack any proteins associated with that. And here we can see our viroids, the ones that are made of infectious RNA only, and they disrupt some of the cellular processes through mechanisms that people still aren't sure about. And here you can see an example on how they result in some really nasty looking plants that are out there. And again, the virioids are only going to be found associated with plants so far. We haven't found them infecting animal cells. And there is sort of a subclass of these, the virusoids, that are this really strange kind of single-stranded RNA that does not replicate by itself. And in fact, in order to go through its replication cycle, it needs to have what's known as a helper virus out there that will co-infect the cells in order to produce more copies of this little guy. And then the last one that we can see out there is these infectious particles that are actually made of just proteins. And if you remember, there is a normal version of this prion protein, the cellular version of the prion protein here, that gets inserted into the membranes of the cells. And when the scrapey version of it that has the beta sheets that makes it really stable, as opposed to the cellular version that has those alpha helices in it, the beta sheet version of the prion protein, the scrapey form, actually is going to run into the cellular form that is on the outside of the cells, because remember, it's inserted into the membranes, and it actually causes what's known as a conformational shift in the cellular version of the protein. So even though they have the same primary sequence with the order of amino acids, the beta sheet form actually causes the cellular prion protein to refold itself, and then you get more of that scrapey version of the protein. And that's going to accumulate on the surface of the cell, and eventually it is endocytosed. And when it's inside of the cell, that's what produces the death of the cells. And if you remember, that cellular version of our prion protein is found highly expressed in the nervous system. So this is going to end up targeting parts of your nervous system and result in death of those cells. And so you get those spongiform encephalopathies with the lesions in the tissue of people that have picked this up. If we go on and start to take a look at the replication of these viruses that are out there, we do find that really interesting kind of steps in the replication of it. And here we see our example with the bacteriophage, which literally means bacteria eater, because that's what they first found when they would add the miracle water from the rivers in India that seemed to cure cholera to a culture with cholera bacteria in it, and all of a sudden those cells would start to disappear over time, and so they hypothesized the cells were being eaten by something that was in the water, and it turns out that was our bacteriophage that was out there. And if you remember, they go through these characteristic stages in the replication. First, you have to have attachment or adsorption as the virus sticks to the surface of the cell, and they're using their glycoproteins to do this process. And this is where we get that host specificity, and not all of these viruses can attach to all the cells that are out there, because you do see that variation in these surface receptors. So once the virus has adsorbed or attached to the surface of that host cell, it needs to penetrate and get its nucleic acid into the cell. And with the phage example, they have to be able to punch their way through that peptidoglycan layer there, and they're going to often use enzymes like lysozyme to digest away the peptidoglycan. And in this example, we see the virus actually is able to punch a hole through the peptidoglycan and through the cell membrane, and that's going to result in it injecting its own DNA into the host cell right there. And once that DNA is inside, it's going to basically co-opt the various enzymes and resources for the cell to make more copies of its own nucleic acid, and its proteins are going to be made. And so that's what we can see here during that biosynthesis phase. Once you've made copies of all your genome and you've made all of the proteins that are going to make up that final virus particle, they do go through the maturation phase, where you're going to see that self-assembly of these proteins around that nucleic acid. And once you've assembled all of those pieces together, now you've got your infectious virion particle, but it does still have to be able to exit the cell. And in this case, the viruses are actually lysing the cell, and that's going to release those new phages that have been made so that they can go on to infect other cells that are out there. And if we look at this process of replication, remember we see this one-step kind of growth curve that's out there. The virus adheres to the cells and injects its nucleic acid into the cells. So that's why we see this decrease in the number of infectious virion particles, because now the virus exists in its intracellular state, where it's inside of the host cell and it actually is not infectious. You go through your eclipse phase as the virus is going to replicate its nucleic acids and make those protein copies during that biosynthesis portion, and then they assemble all those pieces together. And at that point, if you're looking at a lytic virus, then it can burst the cells open, so we can measure the burst time and see how long it takes the virion to actually infect the cells and make new virion particles. And you can also measure your burst size over here. Again, that number of virions that are being released per bacterium or per cell that was actually infected. Remember, there is this other weird example out there that when you look at some of the viruses that are out there, they can go through what's known as the lysogenic cycle. And in this figure, we can see that the normal infection is proceeding right here, but instead of going into that biosynthesis phase, instead the virus is actually going to use some proteins that will insert its genome into the host cell chromosome there. And at this point, the virus is known as a prophage, and it's just going to sit there as a dormant piece of DNA, and every time that cell divides, it's going to get passed on. So effectively, those daughter cells are pre-infected with the virus at this point. And when the cell gets some sort of stress, so often exposure to UV light can do that in the lab, or the cells can run out of food, this actually sends out what's known as an SOS signal from the cell to try and save itself. And that signal also turns out to wake up the virus, and it's going to go back into its regular kind of lytic cycle once the DNA is excised from the chromosome. And then that piece of DNA that's floating around the cytoplasm is going to behave like your normal virus when it's going through that lytic cycle there. They will go through the biosynthesis, the assembly or maturation phase, and then lyse the cells to release these newly made phages to go on and infect other cells that are out there. And there is kind of an interesting little side effect that we see with this process, and it's known as transduction. And this can actually result in DNA being moved from one cell to the other. And what happens is, during that excision process, when the cell is stressed out and the virus pops back out of the chromosome, so its DNA is floating around in the cytoplasm, the virus enzyme actually makes a mistake when it's chopping out the DNA, and it ends up with a little piece of the host cell chromosome attached to the virus genome. And the proteins are going to self-assemble around the outside. It's not like they can distinguish between the two types of DNA at this point. And you're going to end up with a virus that not only has the viral genome, but it can also carry genes from the cells, and it's inside of that virion particle. So this virus is going to go on and infect other cells, and it'll end up inserting that bacteria DNA into the new cell. And so this can actually cause characteristics to be shuttled from one cell to another cell out there. And it's really kind of an interesting process, because remember, our bacteria cells are reproducing asexually, but this way they could actually have an exchange of genetic information between cells, which would be kind of similar to what's going on with your sexual reproduction. It does rely on the process that the viral DNA and the host chromosome, or the viral DNA with the chunk of the host chromosome, has to get recombined with the new cell. And they found that this can actually result in non-pathogenic bacteria picking up toxin genes and becoming pathogenic. And they've actually been able to show examples of this with cholera bacteria from off the coast of India. The virus was floating through the ocean and was able to infect some cholera bacteria off the coast of Chile, so on the completely opposite side of the planet. And those Chilean cholera viruses then became, or cholera bacteria became pathogenic and caused the disease in the people in Chile. I do want to emphasize that when we look at this process and compare it with animal viruses, there are some distinct differences when we go through our cycle there. They can attach and do that direct penetration like we saw with our bacteriophages, but also, and in this example you can see, we have an envelope virus that is able to fuse with the membrane of the host cell, and then actually causes the nucleocapsid to enter into the cytoplasm of the cell. And so there is a difference when you look at this penetration step. But also, as a result of this, these nucleocapsids have to get uncoated in order to release that nucleic acid into the cytoplasm for the cell to start the viral replication going on. So again, at this point, you've got an intracellular virus that is not infectious, and it's going to go through your standard kind of biosynthesis phase there. Often your DNA viruses will replicate in the nucleus, and your RNA viruses will replicate in the cytoplasm there. Once they go through biosynthesis, they'll go through maturation, where they assemble all the pieces together, and you get your new virus, and then they can get released from the cells. Many viruses, again, will use that lysis, where they pop open the cells to release the virions, but also with animal viruses, we can see that process where they're going to be able to bud off the outside of the cells here that we can see, where they actually are taking a chunk of the host cell membrane with them, and those spike proteins were actually embedded into the membrane of that host cell right there. The last thing I want to mention here is that when we look at our animal viruses that are out there, and also our bacteriophages, they can use a variety of different forms of nucleic acids. They can use double-stranded DNA that acts like normal DNA, and they will make their RNA copies of the genomes, or the genes, that'll send messages to the host cell ribosome, which will go ahead and make viral proteins. Single-stranded DNA is similar. It's going to be used to make those RNA copies of the genes. When we look at our our RNA viruses, this is where we see some interesting differences. Double-stranded RNA can exist, and it acts kind of like double-stranded DNA. It's got the messages for the ribosomes to go ahead and translate. Your single-stranded RNA viruses,